Good afternoon, everyone. This is Thomas Kusera speaking here from VarSome. I'd like to welcome you to our webinar organized jointly with Varelas, our distributor for Greece. Regarding the organization of the webinar, the first part is going to be held by Christiana Stanello from Varelas, who is going to introduce Varelas as our distributor for Greece. And then in the second part, I will present our company, and also I will perform a full demonstration of our tools and services for interpretation of NGS data. And in the last part of the webinar, we will definitely discuss your questions. So feel free to send us questions. There is a specific section for questions in Zoom. So whenever you have a question, please feel free to paste it there. And Christiana, over to you, please.
Thank you. Καλησπέρα. Θα ήθελα να σας ευχαριστήσω καταρχήν για το χρόνο που διαθέτετε σε αυτήν την κοινή προσπάθεια να σας προτείνουμε τη λύση της Varelas σε συνεργασία με την Cyprus, την ερμηνεία αξιολόγηση των NGS. Η Varelas φαντάζομαι ότι την γνωρίζετε. Είναι από τις παλιότερες εταιρείες στην αγορά των διαγνωστικών και των βιοεπιστημών στην Ελλάδα. Η δραστηριότητά μας ξεκίνησε το 1978 για τα in vitro διαγνωστικά και το 1997 στον τομέα των βιοεπιστημών. Από το 2008 συνεργαζόμαστε με την Agilent το κομμάτι του Genomics, μετά την εξαγορά της Strategy από πλευράς Agilent. Και στην δεκαετία αυτή που έχουμε διανύσει, έχουμε αρκετά μεγάλη προσπάθεια καταβάλει και νομίζουμε με κάποια σχετική συνεισφορά στις προσπάθειές σας να έχετε αξιόπιστα αποτελέσματα, κυρίως στο κομμάτι των arrays και τελευταία και στο κομμάτι του NGS. Η προσπάθειά μας λοιπόν να προτείνουμε όσο το δυνατόν πληρέστερες λύσεις στον τομέα αυτό μας έφερε κοντά με την εταιρεία Safeto, η οποία ειδικεύεται στα εργαλεία ερμηνείας και αξιολόγησης αποτελεσμάτων NGS. Και σήμερα θα θέλαμε να μιλήσουμε σε συνεργασία με τον συνάδελφο της Safeto για την λύση VarSome Clinical, την πλατφόρμα VarSome Clinical, που είναι μια ανοιχτή πλατφόρμα και η οποία μεταξύ άλλων πάνελ συνεργάζεται πάρα πολύ καλά και με τα πάνελ της Agilent για το κομμάτι του NGS. Σας ευχαριστούμε πολύ και πάλι για τον χρόνο σας. Thomas, you can proceed if you like. Thank you.
All right, Christiana, thank you very much for a nice introduction. We are Safeto. We do bioinformatics. So we are a company based in Switzerland, in Lausanne, and we develop tools for interpretation of NGS data for clinical purposes. And to that end, we have created basically two platforms. The first one, it's called VarSome. It's the one you might have seen before. It's a free platform. You can think of it as a Google of genomics. It's a search engine and community. So what VarSome does is that it aggregates and cross-references publicly available data. And then in turn, it allows you to perform queries against it. I'll show you in a while the real platform. The other platform is called VarSome Clinical, which is already a professional service, which allows you to process and interpret NGS data starting from FASTQ or VCF all the way to the clinical report. VarSome Clinical, it's a complete solution for processing and interpretation of NGS data. You can start either from FASTQ or VCF. So we are a company based in Switzerland, and that's also where we have our own physical server located under our full control. And that's where you, as our client, you are supposed to upload your raw sequencing data, FASTQ or VCF. Once you upload the data, you can start the pipeline. We have a wide range of pipeline covering pretty much all the clinical use cases. For example, we have a pipeline for obviously for single samples, for trios, for couples, for carrier risk screening. We have a pipeline for germline samples, for somatic samples. You can do a comparison between germline and somatic sample, including a pipeline for copy number variation and structural variation. VarSome Clinical runs on our server, which means you don't need to install anything locally. Everything runs on our server and you access it over your internet browser. And at the end of the process, you have the option to generate a clinical report, which may also include links for clinical trials or a list of drugs associated with genes and so on. VarSome Clinical, speaking of sensitivity and precision of pipelines in VarSome Clinical, you can see it's quite high. It's over 99.8%. And these are not like a made up number. These are the results from the precision contest. The precision contest, it's a kind of competition for bioinformatics companies organized by FDA, the Food and Drug Administration Office in the US. And that's where we regularly participate. And these numbers are the results from the latest run of the precision contest. We are 6 years on the market, and we do have already quite a wide user base, client base in Europe. So we have a number of clients in many European countries, as you can see, as well as on the global level. We have a number of clients in the US, in Latin America, in Southeast Asia, Middle East. So we have already a global presence. We have just become a partner with Varelas for the data interpretation part of the pipeline. And what we do here is that we design a bundled solution. The bundled solution means it's the assay bundled with our bioinformatics and delivered as a complete solution. That's what we call the bundled solution. But you can also use VarSome Clinical as a standalone solution for any kind of NGS sample. So with VarSome Clinical, you can process off-the-shelf assay, customized gene panel, exome or genome. It doesn't really matter as long as you sequence on Illumina. And so you can use VarSome Clinical for any of these kinds of samples. Varelas, as our distributor in Greece, is your primary contact in Greece, and is also supposed to provide you the first level of support. But of course, you can always reach out to us or reach out to me if you have further questions. All right, so that's just a very quick introduction to our company and to our products. And now I'd like to proceed and I'd like to show you the real platform. So first, I'd like to start with the free platform called VarSome. So as I have just mentioned before, VarSome aggregates and cross-references publicly available data. And so at the moment, VarSome aggregates over 35 data resources, such as ClinVar, DBSNP, Genome AD, Mondo, Ensemble, RefSeq, and so on. Over 35 data resources integrated and matched together and always kept up to date. And then in turn, of course, you can perform queries against VarSome. So you can perform queries using AGV as term, using chromosomal position, using RSID. You can look up genes, phenotypes, and diseases. So indeed, it functions a little bit as a Google of genomics. And so, for example, here you can see an example of the results for one particular variant in a BRAF gene. So I'd like to guide you through the result page very quickly here. So first, you can see the common information for the variant, such as the chromosomal position, alternate sequence, reference sequence, and so on, and which is followed by a component for ACMG classification. So our interpretation is based on ACMG guidelines, And so in the case of this variant here, you can see that six ACMG criteria fired based on the evidence coming from all these 35 data resources. In other words, VarSome's implementation of ACMG guidelines, it's very solid. It's very robust because it's based on top of all these 35 data resources. It's also very transparent implementation of ACMG guideline, because for each ACMG criterion, you can see here why given criterion fired or didn't fire. You can even click on the blue question mark here and it tells you the details for each particular rule. So it's also a very transparent implementation, allowing you to make your own judgment call if necessary. Of course, not all the criteria can be fully automated. Certain criteria require a patient-specific information or family history information. And so in those cases, it's you who has to make the judgment call and trigger or untrigger specific ACMG criteria based on your best knowledge for that particular patient. And as you trigger additional criteria, you can see that the verdict updates dynamically. On top of that, you can also specify the transcript for interpretation according to ACMG guidelines, which may affect the final verdict, of course. Going further down the page, here you can see the region browser, the gene landscape component here, which summarizes all the variants within a given gene. So there is, for example, quite a few pathogenic variants. There's probably a mutational hotspot here. And for each variant in that region here, you can see the supporting evidence coming from all these 35 data resources, such as ICGC, DBSNP, ClinVar, DBSNP, UniProt, and so on. VarSome counts with currently over 200,000 users, which means there is a growing number of contributions from our global community. So our VarSome users, link publications or classify variants, or start discussions, or even start new collaborations. So there is a growing number of contributions from our global community, which constitutes a unique layer of data, which is nowhere else to be found. And then in turn, we leverage these contributions for certain ACMG criteria, such as PP5. So in the case of the criterion PP5, for example, you can see that it takes into considerations ClinVar entries, as well as contributions from our global community. And we manually check all the contributions coming from our global community. And going down the page, here you can see the list of publications associated with a given variant coming from ClinVar, Civic, PMKB, and so on. PMKB, I'll get back to it in a while. Structural variant browser with all the structural variants coming from Decipherer, DGV, ClinVar, or exact CNVs. So here you can see how well they overlap with each other. RefSeq transcripts, ensemble transcripts, ClinVar entries. So this is a particularly well studied variant. So there's quite a few entries in ClinVar, UniProt variants, allelic frequencies from Genome AD broken down by ethnicity and gender. PMKB, this is a really important somatic data resource. PMKB stands for the Precision Medicine Knowledge Base. It's a manually curated list of variants by Weill Cornell Institute in New York, which also includes the list of drugs and list of clinical trials for somatic cases. ICGC somatic data resource, Civic, genomic data commons, clinical data resource. And at the end of the page, you can see a number of pathogenicity scores as well as conservation scores. So indeed, as you can see on a single page, you get wealth of annotation data And that's why it is so convenient to use VarSome, right? You get all the data from all these 35 data resources on a single page, including the interpretation based on ACMG guidelines. So this is the free platform. On top of that, we have built a clinical platform, VarSome Clinical. So here you can see the real platform for interpretation of your FASTQ files. So the first step consists of uploading of your sequencing data to our server, So first, you have to select your sequencing data and send it over the internet to our server, or alternatively, you can also take the advantage of application programming interface for a fully automated transfer of data. Once you upload the data, you can launch the analysis. You can either start from FASTQ or for VCF. So let's assume we are starting from FASTQ. As I've just mentioned, we have a wide range of pipelines covering pretty much all the clinical use cases. So you can choose between a germline and tumor pipeline for a single sample, or for a family tree, or for a couple, or for a generic multi-sample analysis for extended family, or for cohort studies. Later, you have to specify the assay. So here you can see a number of assays already available, such as true site cancer, or number of assays from Swift Biosciences if you use Swift Biosciences assays. And so if you are using your custom gene panel, you need to provide us the bed file for making it available here in the drop-down list here. Optionally, you can also specify the ethnicity of your patient. You can provide the sequencer details. You shall specify the reference genome, whether it's AG19 or 38. And here you can choose whether you want to call all the variants or only the variants that pass certain quality filters. And here you can also specify whether you want to perform a full analysis or gene list restricted analysis. So here you can specify a gene list. So there are two ways how you can specify a gene list. You can either set it up manually just by copying and pasting the list of genes, or you can take the advantage of HPO, the human phenotype ontology, and set it up automatically. So here you just type in the phenotype of your interest, such as prominent posterior head. And so here you can see, according to the HPO, this particular phenotype is associated with these genes. And so this is the way how you can set up a specific gene list very quickly using the HPO tabs. And then in turn, you can use it. You can use it basically as a filter for your analysis here. So the use case for this option here is pretty much to be able to avoid incidental findings in a larger data sets, such as exomes or genomes. You have to also specify the sequencing data you have uploaded previously. You can also provide sample IDs and sample descriptions here, and you can also specify optionally, again, phenotypes and diseases according to the HPO. And later in the process, you can use these terms for variant filtering and variant prioritization. Once you set up all these details, you can proceed and you can start the analysis. Once the analysis is done, I'm going to show you now a couple of examples. I'm going to start with a whole genome sample. So once this analysis is done, you get the results in the form of the variant table here. So this is a whole genome sample with almost 5 million variants. Variants are sorted by pathogenicity according to the ACMG guidelines. And for each variant, you can see here a number of columns, such as variant name, variant type, column for custom classifications. I'll get back to it in a while. The classification of the of the variant according to ACMG criteria, the list of ACMG rules that fired for each particular variant, the AGVS term, exon number, the gene associated with the variant, mode of inheritance, functions. So in terms of functions, we annotate the variant according to all the transcripts available. So you can see that it's internic, splicing, non-coding, variant, descent, and so on. Zygosity, whether it's heterozygous or homozygous variant. Allelic frequency according to the ethnicity specified in the step before. Allelic variance, which is the percentage of reads supporting the given variant. And coverage, which is the number of reads supporting the given variant. The coverage, you can click on it. It's a link, and so once you click on it, it takes you to the JBrowse view, where you can see the graphical representations of all the reads within a given region. Then around the variant table here, you can see number of tabs with allele frequencies, with clinical data, the list of all transcripts, genes, HPO terms, so for each particular variant, cross-reference samples. I'll get back to it in a while. Nearby variants. And then beneath the variant table here, you can see again, the ACMG classification grid with a full explanation for each particular ACMG criterion, the region browser, ClinVar entries, cosmic entries, caviar entries, allele frequencies, ICGC, somatic data resource, genomic data commands, in silico predictions, conservation scores, a list of publications, and also contributions from our global community, if there are any. VarSome Clinical, it's certified as a CE-IVD device for in vitro diagnostic medical devices. And so it also comes with a number of QC, with a number of QC reports. So for example, here you can access the basic QC report, which summarizes all the details of the analysis. So for example, here you can see the details of the software used for data processing and interpretation, databases used for the interpretation part of the pipeline, summarization of the reads, summarization of the alignment, the number of reads, number of duplicates, and so on, summarization according to coverage, the number of base pairs covered by 5x, 10x, and so on. Summarization according to the classification, the number of pathogenic variants, number of highly pathogenic variants, and also summarization according to each particular ACMG criterion. And at the end, summarization according to the function, the number of synonymous, missense, nonsense variants, and so on. So this is the basic QC report. However, there is even more comprehensive a report called the coding coverage report. So for the coding coverage report, you have to first specify the list of genes for which you want to generate the coding coverage report. And once you do it, you get an Excel sheet with a very detailed coding coverage report broken down by individual genes, region, transcript, exon number, and sequencing depth. Yeah, so this is the place where you can identify the regions with a low coverage or regions that have been skipped altogether. So this is the place where you want to go in case you need to troubleshoot certain issues with sequencing, for example. You can also download the BAM file or index BAM file, and then there's a couple of other options. Now, with VarSome Clinical, basically, you can build over time your private database of classifications and samples. I mean, for example, this is a pathogenic variant, right, according to ACMG criteria. However, if you don't agree with the default verdict for whatever reason, you may reclassify it internally as a benign variant or likely benign variant. And that's the reason why you can see here these tags. Yeah. And these tags stay with the variant forever unless you change it or remove it. Which also means that whenever you upload a new sample having the same variant, you will see the same classification mark here. You can even set up fully customized classifications, such as variant important from a pharmacogenomics perspective, or variant that needs to be discussed with the lab director. And then in turn, again, you can apply these custom classifications to your variants. And so all these custom classifications are private. They stay within your account with VarSome Clinical. And so this is the way how you can build over time your database of classifications. What we can also do, you can also take your manually created list of variants. I guess you might have it, and we can upload it privately to your account with VarSome Clinical. What we also do is that we link all your samples together on the variant level, and that's what you can see here. Yeah. So for example, this particular variant here, you can see that it has been found so far in a heterozygous form in 2 samples, in a sample called exome 7 and in this whole genome sample analysis. So this is something what allows you to go back and forth between your past and current cases, compare the patient phenotypes, and increase the diagnostic yield. And this is what we call the sample cross-referencing. Here, speaking of ACMG, again, you can modify the default verdict by triggering additional ACMG criteria based on your best knowledge for each particular variant. And then in turn, you can save it as a manual classification again. And that's what you can see again here in this column for custom classifications. For example, for this particular variant, you can see that 5 ACMG criteria were modified. So this is again the part of the same story, how you can build over time your private database of classifications and samples. All right, now we have almost 5 million variants, so we obviously need filters. So here on the left side, you can access filters. You can filter your variant based on many criteria, such as ACMG criteria. You can filter your variants based on allele frequency, based on ClinVar classification, based on chromosomal position, based on pathogenicity, based on zygosity, based on function, that is 5-UTR, coding, splicing variant, and so on. Gene list, again, you can apply a gene list as a filter for your analysis, call status, and so on. So just to give you a quick example, let's say we want to filter out only rare variants with a frequency less than 1%. So now we are setting up a filter set. A filter set may consist of several filters, such as allele frequency filter, and the other one can be a filter for pathogenicity, for pathogenic and likely pathogenic variants. So now we have two filters within a filter set. So once you set up a filter set, you can use it all over again for all your samples. So let's apply it. Please note that everything, works pretty fast. We have started with 5 million variants, right? And now we have only four variants in the variant table. So based on the allele frequency filter, over 4.5 million variants have been eliminated, and using the pathogenicity filter, over 330,000 variants were eliminated from the list. And so we are left with only 4 variants. You can always, deactivate certain parameters, certain filters, or reactivate again, and the results updates automatically. You can even go one step further, and you can modify the whole filter set, and you can add additional filter, for example, for PP5 criterion. So now we have 3 filters within the filter set. And again, everything works really fast. As you can see that based on the ACMG filter for PP5, 2 additional variants have been filtered out, and we are left with only 2 variants. So this is the reason why we call these filters dynamic filters, because you can dynamically update any parameter and the results update dynamically according to these parameters. So these are the dynamic filters. VarSome Clinical comes with even more sophisticated filters called algorithmic filters. So algorithmic filters are basically little programs we built specifically for each customer, depending on the specific needs and workflow of given customer. So here you can see several common examples of algorithmic filters. So just to give you an idea about what's possible. So for example, with algorithmic filters, you can do trio analysis, de novo variants for trios, or you can do carrier risk for couples, or compound heterozygous variants, or you can perform a segregation kind of analysis for dominant or for recessive variants, and so on. So these filters can be fully customized according to your needs. Here, for example, you can see how I'm going to show you now exome trio here. So here you can see the original exome trio analysis with 290,000 variants. And then within, you can see a number of sub-analyses. And so each sub-analyze corresponds to the results of an algorithmic filter for de novo variants, algorithmic filter for recessive candidates, and so on. So basically the algorithmic filter creates a snapshot of the original parent analysis. So here again, I'm going to show you very quickly the results. Again, that's pretty much the same dashboard area with a slight difference. So when it comes to trios, we display the zygosity for each individual sample. This is the child, mother, and father, and the same with allelic balance and coverage. Even in the JBrowse view here, you would see the representation of reads for all 3 samples within the same trio. Okay, let me check for questions. No questions so far. All right. So this is and so let me go back to the whole genome sample. So the last missing part is the reporting. So once you filter down the variants in the variant table, you can proceed and you can generate the clinical report. It's your responsibility to decide which variant is going to be reported. So once you decide you want to report this particular variant, you select it for export. And then you can either download the list of variants you decided to report in an Excel sheet, or you can proceed and you can generate a clinical report. So here you can see the reporting part of VarSome Clinical. It's a kind of interactive reporting part. The report can be fully customized according to your branding policy. So of course the logo can be replaced, the header can be updated, you can change the colors, fonts, and sizes. It can be also fully translated into any language. So what basically you need to do is to set up a template, and then you can use it all over again for all your future samples. as I just mentioned, it functions as a drag and drop editor, and so you can control the data you want to include in the clinical report or not. So, for example, here on the left, you can see the list of widgets available for the report. So, for example, you can include the content area, and then you can include, for example, information about the variant, information about the gene, information, the list of drugs associated with given variant, list of clinical trials, and the list of publications. So again, you can decide, right, which kind of data you want to include. So here is the content area where you can provide your specific comment for the report. Here you can see the information about the variant, such as the transcript, AGV ester, the classification of it, gene, exon number, variant type, RSID, zygosity, frequency, coverage. You can control again the data to be displayed in the report. Information about the gene coming from clinical data resources, list of phenotypes according to the HPO, mode of inheritance, list of drugs, a list of clinical trials, and also a list of references. So everything can be everything is fully editable. So, for example, if you don't like this publication, you can remove it, and you can add your own publication, for example. And once you are done with reporting, you can download it as a PDF file or as a doc file. And so here you can see a couple of examples of the final clinical report. Please note this is again just a default template without any specific styling. So you see list of drugs, first information about the variant, information about the gene, the list of drugs, or here you can see a little bit more comprehensive variant, a little bit more comprehensive report, which also includes the information coming from clinical data resources. Here, another example of clinical report, which also shows the list of clinical trials. Something went wrong. I don't know. Yeah. So this is the last step of the process, the generation of the clinical report. So as you can see, VarSome Clinical, it's a complex platform, allowing you to interpret your sequencing data either from FastQ or VCF. VarSome Clinical, it's a certified platform according to the CE-IVD requirements for in vitro diagnostic medical devices. Also, our company is certified with a number of ISO certificates, with the ISO 13485 for data quality and the other one 27001 for data security. Our company is also GDPR compliant, and you can process any kind of NGS data, be it a commercial assay, customized gene panel, exome, or genome. It doesn't really matter as long as you sequence on Illumina machines. Let me check for questions. I think we do have questions now. Okay. Whether we also support data from Ion Torrent? Yes, that's a very good question. So when it comes to the FastQ, it has to be Illumina FastQ. When it comes to the VCF, it can be any kind of VCF as long as it conforms to the standards for VCF files. In other words, yes, VCF from Ion Torrent, PathBio, Thermo Fisher, that's all possible. Whether it's possible to upload BAM files. In principle, it is possible, but there is no user interface currently for it. So you would have to get in touch with us. each time you want to upload a BAM file and send it over email or online storage. So that's a bit more complicated, but in principle, yes, we can also take a BAM file, although all our clients start from FastQ or VCF. All right, I don't see more questions at the moment. Christiana, have I mentioned something? Have I forgot to mention something?
No, I think more or less everything is clear on your side. I guess in case other questions may arise, all the participants can address them either to us or to you directly.
I just got a few more questions here. Whether you can send the files over FTP. Yeah, I mean, from BAM files, I guess that's a good solution. But for FastQ and VCF, you can upload those files directly in the platform using the user interface here, right? So here you just select the files directly from your computer and you send it over to our server. There's a question whether both platforms are free. No, the VarSome.com, this platform, this is a free platform. However, VarSome Clinical, it's not free. This is a professional paid service already, where we charge on a per sample basis. So VarSome Clinical doesn't come with a license. It charges on a per sample basis, depending on the size and type of the sample. So in principle, when it comes to the VCF, the platform charges, on a per sample basis, depending on the number of variants in the VCF file. And when it comes to the FastQ files, it charges also on a per sample basis, but on the basis of megabases in reads, in principle. And Christiana and Varelas will definitely share with you the pricing sheet. There's one more question from Helen. In the list of, if we count a variant with 2 actions, response, no, we don't. I mean, in the variant table here, you can see, the variants that have been called, and genotyped in your data. Yeah. So here you can see the list of almost 5 million variants in this whole genome sample, and so these are the variants that have been called in your data, regardless the classification. All right, so I think we have covered, all the most important aspects of VarSome Clinical. Of course, we are always very close to our clients. We like to provide personalized services. That's the reason actually why we have created these algorithmic filters, allowing us to provide you really personalized filters, specifically according to your needs and to your workflow. I think, yeah. I'm looking at the questions. question from Elizabeth regarding the pathogenicity, how pathogenicity is determined. Yeah, for pathogenicity assessment, we follow the ACMG criteria. Yeah. And then you can use filters to prioritize your variant based on various criteria, such as ACMG criteria or alpha frequency, ClinVar, pathogenicity, zygosity, and so on. So this is up to you how you set up the filter. But we classify the variants according to ACMG criteria. And then we use HPO, the human phenotype ontology, for matching of genes to phenotypes. Yeah, that's the only one we use currently, HPO. Whether we can do CNV analysis, yes, absolutely. We do have a pipeline for copy number variation as well as structural variation. So for example, here you can see this is again the whole genome sample, and there is a sub-analyze for copy number variation and structural variation. So again, here you can see the list of all duplications, deletions, insertions, and so on. And so here you can see the region structural variant browser, where you can see how well the particular structural variation from your sample, how well it overlaps with the public CNVs from ClinVar, Decipher, or DGV, or ClinVar, yeah, or exact CNVs. So yes, we do have a pipeline for copy number variation. When it comes to gene panels, you need ideally 10 samples sequenced in the same sequencing run to achieve the best results for the CNV pipeline. All right, so I think I've answered, or at least I've tried to answer all the questions. Thank you very much for your attention, Christiana. If you have some comment, please, now is the time. If not, I'd like to thank you for your attention. I'll send you the recording for your future reference, and thank you very much for watching.
Thank you. I also would like to thank again our participants for their time, and we will be definitely at their available to discuss any further questions as they arise. Thank you very much for presenting, Thomas, and thank you all the participants for your time.
Yeah, thank you very much and have a good day. Bye-bye.
Goodbye. Γεια σας. Σας ευχαριστούμε πολύ.
Γεια σας.